Ever get the feeling there's like a whole hidden world going on, like just under the surface, invisible to us? Well, there is, inside each of our cells. It's like a bustling city, tons of activity. And today, we're diving deep into the world of actin.
Actin.
Yeah. It's a protein, basically the scaffolding and the highways, even the construction crews of this microscopic city.
Wow, so like without it, our cells would just fall apart.
Exactly.
I've always pictured cells as kind of, I don't know, static. But you're saying it's more like a dynamic, constantly moving thing?
More like a construction project that never might ends, yeah. And these actin filaments, they're the building blocks of everything. But they're not just static beams. They constantly grow and shrink, adding and removing molecules like crazy.
So how do they even keep track of all that? It seems like it would be chaos down there.
Yeah. Well, that's where things get even cooler.
Okay, I'm intrigued.
They have this thing called polarity. Each filament, it has two ends. One's the plus end, and the other is the minus end. And this difference, it's key to how they behave.
So two ends, two different jobs.
Exactly. Imagine like a treadmill. You know how it constantly adds new material at the front?
Right, and it gets taken away the back.
Exactly. That's kind of what's happening here. New molecules are always being added to the plus end, while others are removed from the minus end.
Huh. So it's like this constant flow, like a treadmill, you said.
Yep. And that process, it's called treadmilling. Pretty cool, right? This treadmilling is how the filaments grow or shrink as needed, always adjusting to what the cell needs at that moment.
Wow, that's amazing. But who's controlling all this construction and deconstruction?
Yeah.
It can't just be random, right?
Right. And that's where the regulatory proteins come in. Think of them like the foremen, the supervisors of a construction site.
Keeping everything on track.
Exactly. They decide who gets in, when to build, when to demolish. Like a well-oiled machine.
Okay, so who are these foreman proteins we should know about?
Well, one of the most important is called profilin.
Profilin.
Yeah, it acts like kind of like a bouncer at a club, you know. It binds to these free-floating actin molecules and escorts them to the right spot, the plus end of the filament, make sure everything's orderly.
So it prevents chaos. Otherwise, you'd have these molecules just attaching wherever they wanted.
Exactly. Profilin keeps things running smoothly. Now, for the actual building, we have two main crews, the foremans and the ARP-23 complex.
Foremans and ARP-23.
Yeah, they're the ones attaching to the plus end and speeding up the building. Like, imagine skilled bricklayers just laying down a wall in no time.
Wow. And all of this is happening at a scale we can't even see.
It's incredible, isn't it? But you're right. If you've got all this building going on, you need demolition, too.
Otherwise, it'd be a mess, right? Just a giant tangle of actin.
Exactly. That's where ADF Coughlin comes in. They're like the demolition crew.
Okay. Taking down what's not needed anymore.
Yeah, they dismantle old or damaged filaments and even recycle the building blocks. It's pretty efficient, actually.
So it's all about balance. Building, breaking down, and reusing.
Exactly. And just like in a real city, you need different levels of control, right?
Yeah.
So sometimes you need that whole demolition crew, but sometimes you need something more precise.
Like what?
Like I don't know, imagine a surgeon very carefully making adjustments.
Oh, okay. Very targeted.
And that's where Gelsolin steps in. It's like molecular scissors. It can make clean cuts in the filament, shorten it, or even create branches.
So it's fine-tuning the whole network.
Exactly. That's how the cell can create so many different structures and react to its environment so well. Gelsolin gives it that precision. So we've covered the construction, the demolition, this constant cycle of building up and breaking down these actin filaments, but what are we actually building with all this?
Well, picture this. The cell membrane, it's like the outer wall of our city, right?
Mm-hmm.
But it needs support reinforcement.
Okay, so what? It's like got internal walls too?
Kind of. It's called the cell cortex.
Yeah.
This dense network of actin filaments, that's what gives the cell its shape.
Ah, so the cortex is like the city wall protecting everything inside.
Exactly. But it's not just a static wall. It can actually remodel itself.
Really? So it can change with the cell?
Yeah. That's how the cell can change shape, move around, even engulf things.
Engulf? What do you mean?
Like imagine the city wall just reaches out and pulls something in from the outside.
Okay, that's a little creepy, but I get it.
That's basically phagocytosis. The cell uses its actin cortex to grab things like bacteria or, just like cellular traps.
I was like, the city's got its own little garbage disposal system. That's kind of cool. But we need more than walls, right? Like, what about specialized structures?
Right. Remember those filopodia, those finger-like projections?
Oh, yeah, the ones that help cells move around and explore.
Exactly. Those are basically extensions of the actin cytoskeleton reaching out from the cell.
So if the cortex is the city wall, these Filopodia are like what, sending out scouts?
Perfect analogy. They can sense the environment, even help the cell stick to surfaces.
So they're how wounds heal, right? Cells moving in to fix the damage.
Exactly. And then there's another cool structure, microvilli.
Microvilli, weren't those something about nutrient absorption?
You got it. Think of the lining of your intestines.
Okay, where it absorbs nutrients from food.
Right. That lining, it's covered in millions of these tiny projections, microvilli, to increase the surface area.
So it's like expanding the surface area without actually making the cell bigger.
Exactly. More space to absorb nutrients. And guess what these microvilli are mainly made of?
Actin. Right.
You got it. These tightly packed bundles of actin, they give the microvilli their shape and strength.
It's incredible. One protein, so many functions. But we've talked a lot about structures. How does anything actually move around in this city? We've got to have some kind of transportation system.
Now we're getting to the really exciting part. Let's talk about molecular motors.
So we've got the buildings, the walls, this whole intricate structure thanks to actin. But a city can't just stand still, right? What about the transportation system?
Okay, so think of it this way. Those actin filaments we've been talking about, they're like the roads, the highways of the cell.
Okay, I'm with you. But instead of cars, we have
Molecular motors. And the most well-known one is myosin.
myosin. Isn't that like what makes our muscles move?
Exactly. But it's not just for muscles. It's crucial for all sorts of movement within the cell. Think of it as a fleet of vehicles, each one designed to carry different types of cargo along those actin highways.
Okay, so different myosin vehicles for different jobs. How do they actually work?
Let's start with myosin 1. It's like a tiny delivery truck, okay? It has a head that attaches to the actin filament, like grabbing onto a cable car line.
Okay, I can picture that.
And then it has a tail that can bind to a vesicle, which is basically a container for cellular cargo.
So it latches onto the cargo with its tail and then uses the actin filament to move.
You got it. And just like a truck needs fuel, myosin 1 uses energy from ATP to power its movement.
ATP, that's the cell's energy currency, right?
Exactly. It's incredible how this tiny protein can convert that chemical energy into actual movement.
It's like a perfectly synchronized delivery system, but what are they delivering? Like what kind of cargo?
Oh, you name it. Proteins, nutrients, even larger components like organelles. It's a constant flow of materials, making sure everything is where it needs to be.
So it's all connected. Like even those construction and demolition crews we talked about, they need myosin to get around.
Exactly. Everything depends on this intricate transportation network.
This is blowing my mind. You mentioned myosin is also how our muscles contract, right? What's the connection there?
Well, remember how I said there are different myosin vehicles? Myosin 1, that's our delivery truck. But myosin 2, that's more like a powerful tugboat.
A tugboat. Okay, now I'm really lost.
Instead of carrying cargo, myosin 2 can actually bind to 2 actin filaments at once. And then, using ATP again, it pulls those filaments past each other.
So it's not moving along a single filament, it's bringing two of them closer together.
Exactly. Imagine 2 tugboats pulling on ropes attached to, say, a larger structure.
Okay, I get it. And that's what's happening inside a muscle.
Exactly. It's the combined pulling force of countless myosin 2 molecules that causes the muscle to shorten, which is what we experience as a contraction.
Wow. So all these tiny tugboats create the larger movement of our muscles. That's incredible.
It really shows how much is happening at a scale we can't even see.
This whole deep dive has been amazing. Actin, who knew one little protein could be responsible for so much?
Right. It really highlights how dynamic our cells are. All these intricate processes happening every second just so we can function.
It's truly awe-inspiring. Well, I think we've covered a lot of ground today. Hopefully, everyone listening has a newfound appreciation for the amazing world inside their cells. And remember, next time you move a muscle or see a wound healing, it's all thanks to actin, working tirelessly behind the scenes.